Hi, so this is the third study of Module 1, which is special senses, and today we'll be discussing about the two special senses as well as and they are really combined as one. So we're going to talk about the sense of hearing, which is auditory, and it is also combined with the sense of balance in our body. So we're going to be talking about the eighth cranial nerve today, which is the vestibule cochlea. So there are two names there, vestibule and cochlea. So vestibule means balance and cochlea means hearing. So like earlier, we've already covered the sense of taste, smell, sight, and today we're going to do hearing, and then we also at the same time we're going to do balance. And so this, we said we are not going to cover the sense of touch or position, which is proprioception, that is moving the joint. So before we proceed to the actual sense of hearing, let's briefly understand the anatomy of the sense of hearing. So the organ of hearing is by our ear, and the ear is divided into three parts. We have the outer ear, and so in the diagram you can see the position of the outer ear. So it means this external pinna structure, this cartilaginous structure you see externally covered by skin, and then you also have a canal there that the canal is an S canal. It's not a straightforward canal, but rather an S shape, and then you can see it has an external membranous part and an inner bony part based on the surrounding bony structure, as you can see at the pointer here. So here you have bony part and then you have a membranous part, and the external ear also finishes at the external surface of the tympanic membrane. So the tympanic membrane is also commonly known as the eardrum. So that is where the external ear finishes. So the middle ear is very small, and it is also situated inside the temporal bone, and these you don't have so much air spaces because you are going to concentrate the sound. So when sound enters into the ear, the pinna serves as a funnel for it to collect enough sound and channel it. So when you collect enough sound, like the upper part of a funnel and the narrow part actually directs it. So all the sounds are already directed and by this narrow canal or auditory canal or external auditory canal to come, so that the sound vibration will come and hit on this tympanic membrane, and so that the tympanic membrane is going to move, and when the tympanic membrane moves, it's going to move these small bony structures. There are three bone structures here. So you have the malleus, you have the incus, and you have the stapes. So all of them are going to also vibrate, and these three bones are angled in such a way that they're going to when the tympanic membrane vibrates, the leg of the malleus is going to vibrate as well, or the handle of the malleus, and then it also induces the incus and finally the stapes to vibrate. So the step bone is also going to vibrate on the ears, the structure of the inner ear. So all this, the middle ear is mostly having the bones, and these collectively, these bones are called the ossicles, and there are three of them. And then we also have two muscles at that particular area, and then we also have a narrow canal that connects the middle ear to also to the nasal cavity, and that is why sometimes when the nasal cavity, mostly at the posterior aspect of the nasal cavity, where you have the you have the nasopharynx, so you're going to see a little bit of opening of the internal auditory tube, or it is also called the essential tube. All right. Then lastly, you're going to see the labyrinthine. So the labyrinthine is a combination of the cochlea and the semicircular canals. So the semicircular canals are going to be involved with our balance, the position of our head, that is going to be the flute positions are going to, when our head is standing, these cochlea will sense that our head is standing, and when we move our head to the left, to the side, or whatever direction, these hair follicles or receptor follicles inside the semicircular canals can also direct us to the direction. And then you can see the fibers coming up from the semicircular canal. They're referred to as the vestibular nerve, and they also exit alongside the auditory nerve, which is coming from this cochlea, this nail-looking structure here, which is called the cochlea. And then you can see the auditory nerve. So when the two of them exit this particular, they combine and then go and terminate in the in the pons. They go there together as so you find them mostly at the pontomedullary junction on the lateral aspect, and they are referred to collectively called the vestibulocochlear nerve. So these are the ear and the sense of hearing. The function is mainly to collect the sounds, channel it, and send it into the tympanic membrane, and so that the tympanic membrane will amplify the sound. It's just like connecting your phone to a speaker, and then the speaker amplifies the sounds of your voice. So basically that's what the ossicles does. So they amplify the sound, and then they send in this vibration to go into the cochlea, and these the vibration are going to be interpreted by hair follicles inside the semicircular canal. So what we did was to convert our voice, and then they go into the air, they vibrate the air, and these air tends to make it to become like a wavelength, because it's vibration in the air. And then when they go there, they transform this vibration, then sent in the mechanical vibration is actually a mechanical field. And when they go in, the movement of the tympanic not the tympanic membrane, but the basement membrane of the follicles is going to move up and down, and that moving up and down will also send the hair follicles, which are going to be the receptors, to move as well. So the moment they move up and down, they also transmit this information. So you can see how mechanical information is being transformed now back to electrical impulses by the receptor cells, and these electrical impulses are then sent to the brain, and then the brain is going to interpret this information. So here we're going to talk about it one after the other. We're going to talk about the pinna first. So the pinna is external structure, or we can call it auricle. Is mostly cartilaginous in nature and covered with skin. And then you can also see it serves as a funnel that channels sounds. And then we also have the external auditory canal, and this is the S-shaped canal that we're talking about, that concentrates and channels also these particular sound waves onto the tympanic membrane. And then also part of this outer ear, we have the lateral surface of the tympanic membrane, and we know that tympanic membrane is the eardrum. So let's talk about the pinna first. So the pinna is this particular structure. It is unique, and then the older we get, the more bigger it's going to be. It's not just it's becoming bigger, but rather because the collagen in our skin has now relaxed when we get older, and so it is no more firmed to take a position, but rather relaxed and taking it looking more bigger than its usual position. It has different parts. We can see there are different parts based on the elevations and depressions. And here this one, which one you could touch, and the most exterior fold is called the helix, and opposite the helix or inside the helix you have the antihelix. So you have the helix, the antihelix, and then another important thing is that the helix ends as a lobule. That is where most of us pierce our ears for the females. And then we also, above the lobule, we have what is called the tragus, antitragus. This antitragus is looking at a tragus, and the tragus is just a projection that covers the actual opening of the ear. So the opening here is also referred to as meatus. So anytime you see meatus, it also means an opening. So meatus is not a canal, but rather an opening. All right. So the canal is a longer particular opening opening that is just like a corridor you go through, while the opening is just like a door to it. So that is a meatus. So you have the tragus. So we are not going to talk about the other parts. This is mostly what most of the time what we ask for in this particular program. And then don't forget we have the concha. The concha is a depression just in front of the meatus. So these structures are very sensory in nature, and because they are at the level of our upper jaw, that is why you're getting branches of the maxillary division. All right. So you also have the maxillary division, which is, if you remember, we said is trigeminal nerve. So the trigeminal nerve is because it has three roots. We have the ophthalmic, which is more giving innervation to structures in the orbital cavity and upper part of the nasal cavity. And then we are going to see the maxillary branch. The maxillary branch here is V2, and it is also giving rise to the great auricular nerve, which also innervates the ear, the pinna as well. Then you also have the facial nerve. So the facial nerve is the seventh cranial nerve, and the seventh cranial nerve also comes in from the angle of the jaw, and then sends in different branches. You have the posterior auricular, and then you also have, finally, you have the vagus nerve. The vagus nerve is the 10th cranial nerve, which also have an exterior fibers that goes in to innervate the structures in the inner ear. So the arterial supply of the pinna is coming mostly by the external, the terminal part of the external carotid artery. So the moment it gives rise to the branches of the auricle, and the external of the carotid artery finishes as superior temporal artery in front of the ear. So you can actually see it here. So here you can see that you have the external carotid artery in the front of the face, and just before and the bottom of the lobe you have another branch that goes behind to innervate the auricle from the posterior aspect. This is the posterior auricular artery, and then it goes continues. So the external carotid artery continues upwards and giving rise to tiny branches as well. So you can see one branch going to the lobule, one going to the tragus, and then one going to the upper part of the helix. And then that is why you can see it's inferior, middle, and also superior. So by the time it finished giving the superior, the remaining part of the external carotid artery now is referred to as the superficial temporal artery, which goes to innervate the upper part of the forehead, lateral aspect of it. So the venous drainage also follows also a similar pattern, where you can see the superficial temporal comes down, and also you can see branches from the auricle, the posterior auricular, as well as you have and the superficial temporal marges, and then with some tiny branches of the anterior aspects of the auricle, that is the middle and inferior auricle, will fuse together to give rise to the retromandibular. So when you say retromandibular, it means behind the mandible, and that is why it is called the retromandibular. So when it comes down a little bit, it comes to fuse with the posterior auricular vein and when they fuse with a posterior auricular vein, they also fuse with some other vein, deeper vein, which includes the which includes the the pterygoid plexus. So branches from the pterygoid plexus, which are draining the nasal cavity, assist in draining the nasal cavity, and parts of the oral cavity also, they give rise to branches will unite with the retromandibular, and when they all unite together, they form the external jugular vein, and downwards the external jugular vein and the internal jugular vein will fuse together to give rise to the superior vena cava, which empties into the right atrium of the heart. So now we're going to talk about the external ear canal, or So the external ear canal or the auditory canal is not a straight shaped structure, but rather is an S-shaped structure, and that is why when you try to look into it, you do not see it, especially if you're trying to observe the tympanic membrane. What you have to do is you have to pull the lobule of the ear, and then inferiorly, and then you pull it towards you. That is when you tend to see that the S-shaped canal is more straight, and thereby you could be able to focus and see the tympanic membrane. So like we said, it's S-shaped, which is the lateral 1/3 is more cartilaginous, while the medial 2/3 is bony in nature, and it is situated in the temporal bone like we mentioned earlier on. So it is also lined by skin. So the skin also contains hair follicles. So you remember when you so you have squamous epithelial tissues here, and they also have modified sweat glands that produces the ear wax. Those wax are important because they help to trap bacteria and dust, as well as tiny insects coming into the ear. So you do not necessarily need to cut off all the hairs in the ear, because they help protect against foreign particles. So the function is to conduct sound into the ear, and also it receives a similar arterial supply like the pinna. It also gives rise to some of those branches coming in, especially where you have the middle, superior, middle, and inferior auricula. They also extend some branches into the canal to supply the canal. So when we talk about the innervation, it also receives various innervation, including the facial, which is in yellow. It also receives the auriculotemporal nerve, which is a branch of the maxillary branch of the trigeminal nerve. That is the one shown in green, and in yellow you can see that is the facial, coming from the chordae tympani. And then you also see the purple one is coming from the glossopharyngeal nerve. So the glossopharyngeal nerve give rise to the auricular. You also have you also have some parts giving rise to the vagus nerve. So here we're going to talk about the middle ear. So the middle ear is composed of the inner surface of the tympanic membrane, and then you also have the bones of the ossicles. So you have the malleus, the incus, and the stapes, which you can actually see them here. So they've been magnified so that you're able to see it. The foot of the steps is also very important because it closes an oval window here, which tends to communicate, which the stamping of the foot sends vibration into the cochlea, and then the vibration of the cochlea and the fluid here will give rise to different signals. So tympanic the tympanic cavity, which is the middle ear, the structure, the part of the middle ear, is also lined by mucosa, and also the inner surface of the tympanic membrane is also lined by the mucosa. It contains the different bones. Like we said, we have also two muscles, which are very tiny muscles. So stapedius is the smallest muscle in the whole of the body, smallest striated muscle. We have the tensotympani muscle, which also attaches to the foot of the malleus, and then extends all the way to the tube, the walls of, and insert onto the walls of the essential tube. So the tympanic membrane, we said it has an external surface and an internal surface. So the internal surface is the one that has the hands of the malleus, and then the outer surface this is a view from the outer surface, and so it's very transparent but very elastic, and it's also very the lateral surfaces are thicker than the middle surface, you can see, and so that is why the middle surface is more transparent, while the lateral surface or the brim attaches to the bony walls around the tympanic cavity. So like we said, the tympanic membrane common name is the eardrum. It is cone in shape because of the pull by the hands of the malleus. It pulls it, so it gives rise to a slight cone shape. You don't want the tympanic membrane to be straight, because when it is straight it is easy to tear. But when you have something a little bit corned or pulled away, what happens is that you reduce the surface tension. So that is what the surface tension is not just at the middle, but rather now you also have something supporting the middle aspect of the cone. So that is why you tend to have less tendency of tearing of the tympanic membrane than when it is smooth. So it is separated. It separates the middle ear and the outer ear and the middle ear, and it has three layers and transits information. So from the air, and then it becomes a mechanical structure from the air vibration, and then it becomes mechanical because of the movements of the bones of the ossicles. So we also have, so outer layer is also lined by epithelial tissues, and then you have the middle layer, which is very flexible, and it's made of the fibrous tissue, which also contains blood vessels. And that is why, and the blood vessels is mostly found at the periphery but not at the middle aspect. So that is why when you have a tear of the tympanic membrane in any kind of accident, be it a loud voice, or maybe you put a foreign object into the ear and it tears the tympanic membrane, it takes a while for it to heal because of the lesser blood vessels found in the middle aspect of it than the lateral aspect of it. And so the inner layer inner layer is made of the mucosa. So the mucosa is the layer that is more in towards the middle ear. So now this is the tympanic membrane. So this is mostly the view from the exterior. The view from the exterior, you also be able to look at it. So this illustration, this is the one on the left hand side is a schematic illustration, while the one on the right hand side is an actual picture taken during an assessment of the of the ear of an individual. So here you can see what is called the umbo. So the umbo is the joint or the last part at which the hands of the malleus attaches to the tympanic membrane. And then you can see a bright light here. That is mostly when you flash a light to observe the inner ear, you see this cone light. So this is what you see it here around this particular aspect. So you can appreciate it in the anterior superior quadrant, because here we can divide it into four quadrants. So here you have the posterior superior quadrant, you have the anterior inferior quadrant, and then you have the posterior inferior quadrant. It has been separated so that you'll be able to appreciate that. So here you can actually trace a little bit of the malleus, and then at the base of the malleus you also have the incus, which is now clearly represented in this image. However, you can see this particular folds where you see this posterior and anterior malleolar folds, so you can actually appreciate them a little bit on, and that is, so in this posterior malleolar fold is more prominent than that of the anterior malleolar fold. All right, so that is what you get, what you see there. Yeah, they're also very important. And then you can see that different parts, so more of the anterior aspect is innervated by the so this is V3, that is the mandibular division of the trigeminal, and then you also see the facial nerve, and then you can see auricular branch of the vagus nerve. So these are doing the sensory innervation of the tympanic membrane. So now we start talking about the middle ear. So the middle ear, like we've been emphasizing, is made of three bones that are very tiny, located in the in the tympanic cavity, that is the space there. So here you can see the bones together, but when you separate the bones, And you can see malleus. So this is the handle of the malleus, which we saw earlier on, attached to the tympanic membrane. And then you can see the head of the malleus, which is going to attach to the incus as well. And then you can see incus has two processes, and one process is going to attach to the stapes. And then you can see the foot, the stapes has two foot, two processes that attach to a foot, which is very important. So here the malleus is also referred to as the hammer, and then you can see the incus is also referred to as the anvil, and then the stapes is referred to as the strips. So the both collectively functions to amplify and transit the sound vibration into the inner ear as mechanical information. So we're going to talk about the malleus. Malleus is the largest bone of all the three. It has the head, the neck, and then it has the handle, which is also referred to as the manubrium. And then you can see it has two processes. It looks like a baby with one leg trying to hug you with two hands. So they have the anterior and lateral. So this is the anterior and this is the lateral and processes. And then it also, you have the head that articulates with the incus, just like we said earlier on. So here you're going to see this is a magnified aspect of the incus bone or an anvil bone. So you can see it looking like that. And then you can see this is the body. You have a short process and a long process. That, like we said, mentioned earlier on, the long process connects to the head of the stapes bone, and then and so that is one of the significant aspects of it. And then you can see the stapes bone is the smallest bone, and it also has the smallest muscle, striated muscle of the body. It has a head, okay, like it's going to the head is going to attach to the long process of the incus. And then it also has the neck, and a very narrow neck, and then it has anterior and posterior cruces, or the processes for this anterior and posterior cruces. And then you have the foot of it, or the foot plate. So the foot plate is the one that covers the oval window and the in the in the inner ear. So so you can see it it fits onto the oval window of the inner ear. And then the muscles, we have two muscles. You can see this is the longer muscles, and then because you don't want it to have a very, you want this muscle assist the hand on the hand of the malleus, to prevent the flattening of the tympanic membrane, to give you that slight curve or funnel shaped. So that is what the malleus does. And so the malleus does that in control of the tensor tympani muscle. That is, the tensor tympani attaches to the hand of the malleus and goes into the wall of the essential strip to to attach. So what it does is, like we said, reduce the vibration amplitude. So when there is a high sound, sometimes before you even put your hands to block the sound, the high amplitude of the sound, also the tensor tympani is important because it tries to reduce that. It is innervated by the medial pterygoid nerve, which is also a branch of the mandibular nerve. So the mandibular nerve, medial pterygoid is a branch of the maxillary nerve, not the mandibular nerve. That is a mistake. So here we have the smallest muscle, which is the stapedius. You can see it's very tiny. It is the smallest skeletal body, and what it does is reduces the vibration of the stapedius into the walls of the inner ear. So what you're viewing here is you're viewing the middle ear via the inner ear. So the inner ear has been removed. So it's just like you going through the skull. You remove the brain and then you go through that side to appreciate this tympanic membrane. And then you can see the foot, the foot of the hand of the malleus, and then you can see the tensor tympani muscle, and then also you can see that the stapedius also attaches to the posterior wall of the middle ear. And what it does is also functions to protect the inner ear from loud sound. If you need to reduce the fast phase of the vibration of that. So this muscle is innervated by the facial nerve, which is coming from the quadrant tympani branch of the facial nerve. So here we have the essential tube. This is a tube that runs, or we could also call it the auditory tube. We could call it also the pharyngotympanic. Reason pharyngotympanic because it opens into the pharynx, but also opens into the tympanic cavity. So pharyngotympanic tube, and also it is important because it tends to balance the tension or the air pressure between the external ear auditory tube and the middle tympanic cavity. So the auditory tube helps in reducing that pressure when you're flying up or changing the altitude. Wherever you're changing your altitude, you tend to feel like there is a pressure inside your middle ear. Sometimes you need to cry, or sometimes you need to chew chewing gum, so that you constantly open your mouth and open your as well as the nose to reduce the pressure in the middle ear. If not, it can actually break the tympanic membrane. So the key functions of the key features of the essential soup is that it has a bony portion. So the inner third of the tube is bony, so you can see it in that particular aspect. But as it comes towards the pharynx, it becomes more cartilaginous. That is why it is called the outer two-third tube, which is cartilaginous and more flexible. It opens into the pharynx. That is why it is called the pharyngeal orifice. So you can see it in the the nasopharynx aspects of the that is behind the nasal cavity, inside opening at the pharyngeal aspect of the one in the nasal cavity. That is why it is called the nasopharynx. And then also you have the tympanic orifice, opening of the tube into the middle ear. It's also referred to as the tympanic orifice, or you can also refer to it as the essential tube orifice. So the arterial supply is coming from the maxillary branch, okay, maxillary artery, and then you also have the posterior tympanic artery, which is supplying the posterior aspect, and the mastoid air spaces. So the air spaces surrounding the middle ear cavity or the tympanic cavity is called the mastoid air cells, and these are also very important because they helps in sieving or reducing and the intensity of the sound sometimes. So you have the stylomastoid arteries are also is a branch of the posterior auricular arteries, and these posterior auricular arteries, you remember, they are coming from coming off from the external carotid artery, and they supply stapedial muscles as well as the posterior wall of the tympanic cavity. So we have the venous drainage is by the pterygoid plexus. If you remember, we talked about the pterygoid plexus, and so the pterygoid plexus and the inferior petrosal sinuses that drain into the brain, and then from there they drain into the sigmoid sinus, and from the sigmoid sinus they drain into the internal jugular vein, and then finally into the superior vena cava and to the heart. So the sensory innervation is coming from primarily from the tympanic plexus and lesser petrosal nerve. So these are branches of the glossopharyngeal. Earlier on we talked about the glossopharyngeal also innervating the anterior part of the tympanic membrane. So here we also see some branches as well, the tympanic plexus as well as lesser petrosal nerve. They are also innervating the walls of the and they are innervating not just the tympanic membrane, but as well, that is the anterior part of the tympanic membrane, but as well they also innervate the walls, that is the mucosal lining of the mastoid ear spaces. So you also have nerve innervation, that is the quadrant tympani. So here this is so you can see this is the quadrant tympani, and this is very important because this is a branch of the facial nerve. And also you have the auriculotemporal fibers coming from the V3, as well as you have the vagus nerve as well. So clinical application of the middle ear include the otis media. So you could have an acute or a chronic type. So the acute type is more common in children because of infection, and in symptoms include the ear pain, fever, and hearing loss, temporal hearing loss. And then you have the chronic otis media, which is a persistent and recurrent middle ear infection that can lead to hearing loss, ear drainage, and potential complications. So I think ear damage, that is what I wanted to write there. And so most of the time you get to see sometimes you get to have coming up from that particular aspect. Then we also have the otosclerosis. So anytime you hear otosclerosis, it means a breaking down. So this is associated with the hereditary bone diseases that cause progressive hearing loss because of the breaking down of the mastoid ear spaces inside the middle ear. Okay, and sometimes they extend onto also to the bones. So the bones are also going to the bones of the ossicles are going to be attacked and be degenerated, and thereby they cannot function, so vibrate to produce or amplify the sound. So we also have the tympanic membrane perforation. So that is where you injure yourself, or accidentally or on purpose. And so how to diagnose, we use the otoscope, and so this is where you can actually view using an otoscope to visualize the tympanic membrane. You could also do other type of clinic analysis. You can also use a tympanometry, where you'll be able to check the mobility of the eardrum in response to air changes and pressure. And there are other several ones. You can also do an acoustic reflex testing to assess the contraction of the middle ear muscles in response to loud sound. So you want to see whether the tensor tympani and the stapedius muscles are functioning. You also want to do the auditometry measurement, where you are trying to check for the hearing sensitivity at the different frequencies as well. So that is mostly for the inner ear. So the also clinical application regarding the essential tube is you also can have a dysfunction of this tube, where it is associated with allergy or respiratory infection, mostly associated with the upper part tract of it. You could also have an otis media infection, and also so you can also do several treatments by maybe decongestants. Nasal corticoids are very important. They're used to help in reducing mucus production as well, and etc. So these are also another set of clinical application associated with the essential tube. This is sleep disordered breathing. This is very deadly, because sometimes because of the drop in the position one sleeps, you might end up having different type of blockages. Some blockages could be some of the blockage could be associated with the essential tube, and thereby also resulting to deep breathing difficulties that could lead to death. So this is called the sleep apnea, and it is associated with both the pharynx conditions associated with the pharynx. So now we're going to be talking about the middle ear. So the the inner ear. So the inner ear, we said, is a very complex structure, and it's made of two different type of apparatus. So this apparatus, we have the cochlea and the vestibular systems, and both of them form the its cranial nerve, which is vestibule cochlea. So this structure, we have an external bony structure, and inside it is just like a tube surrounding another tube. All right, so our outer tube is bony and the inner tube is membranous. So here you can see this is the bony aspect of it, and that is the membranous aspect, which forms the ductal system. So here you can see you have a coiled one that looks like a snail, that is cochlea, and then you can see three of these semicircles. They are not complete, and that is why they're called the semicircles. So there are three of them, and then you also have an ampulla at the base of it. They have ampulla. Each of them have one ampulla. They don't have at the two ends, but rather at just one end. So they have an ampulla, and this ampulla is very important because it contains also some hair cell receptors, which we're going to see. And then you also have the utricle, and also you have the saccula, and then you also have that particular aspect, which is the cochlea, which is also externally you have the bony structure, and internally you have the membranous structure, which forms the canal. So the junction at the end of that snail-looking structure of the cochlea is called the helical. So here we're going to talk about the bony and membranous labyrinthine. So what it means is that the bony labyrinthine is a structure that covers the external aspect of it. So here you can see this is the bony aspect, where this transparent structure is showing you the membranous. So the blue structure is the membranous aspect of these particular complete apparatus as well. So when you look at the bony apparatus, this is where you have the cochlea, and this is where you have the round window, and this is the oval window. The oval window is where you have the foot of the stapes. So the foot of the stape is pumping here, and the fluid inside this membrane. So the fluid inside the membrane are the ones that will be just like if you have water in a cup and you keep hitting the top of the cup, you see the water is vibrating based on the way you're hitting the cup. So that is exactly the information, or the vibration of the fluid, and exactly the sound, the amplitude of each vowel we speak. That is exactly how the fluid inside the membrane is going to vibrate, and that vibration, or the moving or causing the fluid to become wave in nature, the movement of the fluid inside to form that wave also moves the hair follicles in the inner ear, and these movements are also, they generate impulse, action potential, and this action potential are the ones that are being sent to the auditory centers of the brain, which is mostly in the tympani, the temporal loop. All right. So here we have the red is also showing the membranous labyrinthine, and then you have the blue one is the bony labyrinthine, and just like we explained earlier on. So here we're going to start talking about the cochlea. So the cochlea is the part that is involved with the auditory or hearing, and then you can see that it starts from the base or the lower aspect of the saccule. So it goes around almost 2.5 times. So you can see this is one, and then you can see two, and then you can see a tiny one here. So that is why most of the time we say that the cochlea is a snail-looking structure that goes like 2.5 times in a cycle. And then you can see that at each base of this blue structure, or the blue membranous labyrinthine, you can see some fibers. So those are the afferent fibers that are leaving the receptor cells. They are going to exit and collect at the midline, more as the auditory or the cochlear nerve. So these bony labyrinthine and these membranous labyrinthine are also divided into two. It is going to divide into two chambers. So you're going to have So you're going to have one on the bony labyrinthine. Because of the membranous labyrinthine, we're going to have two chambers. You're going to have one on top, one below, and then you have the middle chamber. So the middle chamber is the one that contains the organ of Corti, which means the organ of hearing. So then both of them are going to move, and when they move, they create those impulses that goes there. So I know we you're confused. This is what I'm going to explain. So if I cut this area and bring it here, what you're going to see is that this is the membranous labyrinthine you are having, and then you have the upper part. You're going to have the bony so you're having the bony labyrinthine. So here you see the bony labyrinthine outside. So when you get to appreciate the bony labyrinthine, then you have a membranous labyrinthine inside. So the membranous labyrinthine in the middle contains the organ of Corti, and the upper part of that, okay, is referred to as the scala vestibule, and the lower part of it is called the scala tympani. So this is very important because when the fluid are moving, we are going to have fluid on top and on below. So the fluid and then you also have fluid in the middle. So and these in the middle, like we said, is the contains the organ of Corti, or the organ of hearing. And then, like we said, we have the scala tympani, which is also containing a fluid which is called the perilymph fluid. And then you have the scala media. The scala media is the one in the middle. It contains the organ. It contains the cochlear duct, or it is also referred as the cochlear duct, and it contains the otoliths. Okay, and then you have the scala tympani, which also contains the perilymph. So these perilymph and the two perilymph, that one and the scala vestibule, it takes the first vibration. So when the foot of the steps vibrates on top of the oval window, the fluid in the perilymph fluid in the scala vestibule is the one that vibrates. When it vibrates on top, it also sends this particular roof and as well these particular membrane. So we're going to magnify it, and then we're going to see the different parts of these structures, and then we're going to see how they move when the fluid, the perilymph here and the scala tympani, and also vibrates. So when it finishes vibrating, it's going to vibrate all the way in the superior aspect, and when it comes to the tip of it, what happened is that it's going to have a contact fluid, so it's going to continue. So the helicotrema is the point at which everything terminates. All right, but however, it's scala tympani is also containing fluid, which also it takes excessive voices or sound, okay, excessive sound out of the inner ear or the cochlea, such that it reduces damage in that particular area. Okay, so now here we can see it is being magnified, and then you can see we can appreciate the organ of Corti properly. So here you can actually see the vestibular membrane. The vestibular membrane is important because when the fluid is vibrating here, it's also vibrating. And what happened is that if you can see, the vestibular membrane is also attached at a particular point. And what happened is that when it moves, this particular the fluid here moves, the vestibular membrane moves as well. When it moves, what happened is that because it is attached to this particular roof of these the organ of Corti, and so this particular structure is going to move back and front this way. So it's going to pull this particular structure up front and back, and then when it moves this particular structure front and back, what happens is that there are tiny hair follicles, there's receptor cells here. So you have one row of receptor cells here and 3 rows, 1, 2, 3 rows of receptor cells. However, audition is more, or perception of sound is more by the first hairline receptor cell. So this role is called the first role, and they do most of the information, like 80 to 85% of the interpretation of the sound is being taken up by this first role of hair the inner hair cells, receptors, and then you have the outer. So 1, 2, 3. You have 3 outer hair rolls, and they also. But even when this tectorial membrane moves, it also moves across all the hair follicles. But like I said, the inner one, this single roll, is the one that does majority of the function of hearing. And then you can see it is containing the nerve endings are very close towards the point of exit of the cochlear nerve. So at this image, we are going to see this is the magnified aspect. So you can see the scala vestibule, the scala tympani, and also you have the scala media, where you have where it's also called the cochlear duct. And then you can see that this is the vestibular duct. It's also called the vestibular duct. You can see every one, because it's snail shape. So we can just cut one. This look at it is a snail shape. So this area has been magnified, and then you can see that all of them have in the exit of the cochlear fibers at the axons of the cochlear nerve. So here, this particular area, because of the cochlear duct, or where it contains the organ of Corti, is being magnified. And here you can see the tectorial membrane. So you can see 1, 2, 3 hair cells. So these are the 3 outer hair cells roll, and this is the inner hair cell roll. And what happened is that most of the time when the foot of the steps is being hit, the perileaf inside the scala vestibule is the one that vibrates. And when it vibrates, what this particular vestibular membrane also pulls. So it is located at this point. When it pulls, movement, like, because it's vibrating, it's going up and down. So what it does is also moving the tectorial membrane, going right and left, right and left. So the intensity of the sound also varies how it moves. The speed at how it moves, distance, and everything is being calculated to exact sound that we make. So these impulses, or the movements, this intensity of the movement of this tectorial membrane sends the impulse, so it moves the hair follicles, which generates action potentials, and these action potentials are carried by these inner hair follicles, and then these nerves, the afferent nerve at the base of these inner hair cells, which are the receptor cells here, takes the information and they exit the inner hairs, the inner ear. So this is just a description. You can pause it and read these different types of discussions we did, but I've already explained these hair follicles, and we can see that they're all you can pause it and read. And then here is the point at which how sound is being detected. So when we have a sound, it moves, is vibrating as a sound waves, and these sound waves comes to hit on the tympanic membrane, and when the tympanic membrane hits, the hand of the malleus is also being vibrated, the head vibrates, and this head vibrates tends to vibrate the head of the incus, and then you can see the long process of the incus is being vibrated, and then also this sends the vibration to the crus, the right and left crus, anterior and posterior crus of the steps, and then you can see the foot of the steps is vibrating over the oval window, which opens onto the perileaf. This is the vestibular, the scala vestibule. So the perileaf of the scala vestibule tends to vibrate, and the thing is, the information are being taken up at different points. So the loudest, so you can actually appreciate that the different amplitude or sounds are being taken at different parts, and then you can see that when information when the vibration finish, excess sound is being taken away from it via the perileaf inside the scala tympani. So you can see this is going out via the scala tympani and goes all the way to the round window. So this excess sound is moved out of through the round window. So you can see that the highest amplitude is taken up immediately. It starts to vibrate, and then you can see that the ones with the lowest frequencies are the ones that are being detected at the tip, almost towards the helicotrema. And then you can see at the bottom here, what you see in blue is the exit. Okay, so it is going to take you back to the base, and the base is where you have the round window. So you can see information goes into the oval windows, and then after it finish interpreting, excess sound is taken away where the scala tympani, and then they go out, they exit via the base, that is the round window. So you can see it's telling us that B, in this image B, you can see the high frequencies are taken up immediately when it enters the cochlea, where low frequency middle frequencies are taken up in the middle, maybe here, and low frequencies are taken up around closer to the helicotrema. And so that is to tell you how these sound frequencies are being interpreted at different parts of the cochlea. And so this information, when this when this information goes there, at each part where this information gets to vibrate and then send the impulse through the hair, the inner hair cell receptors, this information gets to the afferent fibers, and from the afferent fibers they move into they enter into the they ascend as the first order neuron, and then they go to from the cochlea, they go to the cochlear nucleus, and the cochlear nucleus is found at the pontomedullary junction. And then you can see that afterwards they ascend to the superior olivary nucleus. Superior olivary nucleus is also found at the medulla oblongata, and then you can see some fibers crosses to the opposite side, while some of them remain in the same side. So the ones remaining in the same side is called the ipsilateral, while the ones crossing to the opposite side are contralateral. This is because we want to avoid where in a situation where ear is faulty, what happens is that we want both the side of the brain to be able to interpret this information, or sometimes when one side of the brain has a lesion, it means that we can still understand, we can still be able to hear. So you can see that the first order neuron is the one that comes up from the cochlea and comes into the cochlear nucleus in the on the pontomedullary junction. It enters the superior olivary nucleus in the medulla oblongata, that is the second order neuron, and a third order neuron enters into the inferior colliculus. You remember, inferior colliculus is situated posterior to the midbrain, and then from the inferior colliculus it goes into the medial brachium and goes into the medial geniculate body. So the medial geniculate body is a part of the thalamus, which is located behind the thalamus. If you remember, we said we have medial and lateral geniculate body. The lateral geniculate body is going to be taking information from the vision, and then the medial geniculate body is taking information from auditory. And so from the auditory, it also serves as like a thalamus, which is a relay station, and then from there it projects into the final or primary auditory cortex, which is at the temporal lobe. So you can see the first order neuron, the second order neuron, the third order neuron, the fourth order neuron, and then finally it moves from the geniculate body, it moves into the primary auditory center and the temporal lobe. So you can see it here at the temporal lobe, which is the auditory receiving centers. So both the primary and secondary auditory centers. So now we're going to talk about the vestibular system. So the vestibular system is the one that deals with the semicircular canal, the utricle, and the saccule. So you can see them in this particular image. So they are involved in equilibrium, that is balance and body spatial orientation. So when you are standing or jumping, you actually know which position you are. So the fact that we subconsciously know the position our body is because we have this spatial orientation due to our equilibrium, and most of this is calculated by the position of our head. So the position of our head helps us in the orientation of our body. All right, so for detecting motion, head position and spatial orientation. That is what the vestibular system does. So also it's involved in motor functions by triggering reflex motions to the main stable or restabilizing equilibrium. So if you miss your steps, what happens is, because you miss your step, your body also helps you in jacking because of the head position. If your head moves away, automatically your body is trying to pull back your head, and you have to be in the midline. Your body wants to streamline everything to be in the midline. So also perception of gravity and movement, because of the your hair follicles are also going to be working with gravity as well. And also if movement up and down, front and back, and also it's all because of the orientation of the hair follicles, which are also situated in the ampulla and the utricle, which you're going to see. Also because they also helps in stabilizing the head and body during movement, and also maintain our posture. Even when we're driving a car, if there is somebody that steps on a brake, what happens? Our head moves, but our body tends to pull back our head to reposition it. So all these are parts of the vestibular system. So, the vestibular system also has a bony labyrinthine and a membranous labyrinthine. Is also filled with endoleaf, and also you have the three semicircular canals. So these three semicircular canals, you have the anterior, you have the lateral, okay, and then you have the posterior. So you have anterior, posterior, and lateral. So these are you can see the positions. So most of the time anterior and posterior, they meet at the midline, and then lateral, you can see it having its own base. So you can see that instead of having like six endings, they are instead having three ampulla areas, and then only two ends, other ends. And then you have another and you have another structure which is called the otolith organs. So the otolith organs are important because they detect the motion, or either we are moving horizontally or vertically, and helps us in assigning the position of our head. So these otolith organs are called the utricle and the saccule, because they also have those receptors in them. So here we're going to be talking about the ampulla. So the ampulla are associated with the three semicircular canals. So you have the anterior, posterior, and lateral semicircular canals, and they contain sensory receptor cells, which are also hair cells. So when you magnify when you magnify this at the ampulla, this is exactly what you see. You also see hair follicles, and these hair follicles are also, because you also see the receptors. These are having the receptor cells, and then you can see the nerve endings at the base here. So on top of the hair follicles, you have what is called the fluid there, and this is called the endoleaf fluid. And the movement of this endoleaf will also tends to move the position of the hair follicles as well. And the movement of this hair follicles tends to induces action potential to release neurotransmitters, and those neurotransmitters are the one that induces the sense information to the brain. So when you come to the otolith organs, you have this the utricle and the saccule. So in the utricle and the saccule, you have something slightly different from what you have at the ampulla. So what you have is that you also have a fluid, you have an arrangement of different types of receptor cells, and you also have the supporting cells. You can see the arrangement. And we have two types of cells, receptor cells, but we are not going to dive into it this thing. So here you also have a certain type of hair follicles, and these hair follicles, unlike the one you saw in the ampulla, the hair follicles are arranged in one particular direction. It's not curved like how you saw them in the ampulla. And then you also have what is called the odontoconeus. The odontoconeus are crystal shaped looking structures that actually help to support the superior weight of the fluid, or the odontalic, that is the fluid surrounding the hair follicles. So here we're going to be talking about the ampulla, and the ones in the ampulla are important because they are they function. The movements of the fluid also function in the direction of the head. So the semicircular canals, you can see what it does is it tells us the position, and the anterior one tells us when we flex our head to the anterior aspect, it gives us this particular information as well. And then when you have the lateral, it gives us side to side movement. And then posterior is also like if you're going to flex our head right and left as well. So for the anterior is front and back, and flexion of the head up and down like you're nodding. And then the lateral is if you're you're standing at 90 degrees, you turn your head like saying no no no, like that. And then the posterior semicircular canals also tells to interpret the information. If we tilt our head left to the right, that is what it does. So here you can see this is the anterior doing that. So you can see the movement of the fluid when you flex your head to the up and down. This is what the information, the fibers also do. The fluid tends to move as well with the hair follicles, so it tells you the direction, the position where your head is. And so that is why when sometimes when you bow your head for a long time, if it's a very very long time, you start, when you put it back, you start to feel dizzy because it has been away from the natural position. And so when you're using lateral, you say no no, this is how it moves, and that is the information of the ampulla. It also moves this at right and left, it moves right and left as well when you move your head. So here when we talk about the otolithic organs, so the otolithic organs are, we said, utricle and the saccule. They also detect the movement of the head, forward and backward movement, and they also tend to move in gravitational force. So gravitational force, if you're going against gravity, let's say for instance you are in a lift going up and down. Sometimes when you enter a lift where you feel, or when a plane is taking off, there is a pause, you tend to feel like your head is pressing backwards. These informations are also important. And when you had a head-on collision, if you're moving, or not a collision, maybe let's say you are in a car and the car is moving very fast, and then somebody matched the brakes, what do? You move backwards, right? Your body tends to pull your head backwards. So those are these the functions of the otolithic organs, that is the utricle and saccule. They tell you the position. So the utricle is it interprets information in a horizontal plane. So for instance, if you're moving and then you press a brake, what happens? You come back. Okay, your body, you just feel like your body jacked you and you came back. So that it is a function of the utricle, while the movement of a vertical plane is done by the saccule. So the saccule is the one that does, if interprets the information if you're going up and down, or if you're taking a plane or taking a lift. So this is the anatomy of the structure. So these are the otolithic crystals, and then the fluid, they are there to suppress, to prevent and the excessive movement of the jelly structure. So the crystals are there to make it move gradually, and also when they move gradually, what happens is that they change the position of these hair follicles, or serocilia, or microvillar cells. And when they tend to move, it induces an action potential by the cells, and these action potentials are taken up by the nerves, and these nerves endings go in or exit this particular inner ear area as the vestibular nerve, and then they go to interpret the information. So because they are also doing balance, they are going to communicate heavily with the cerebellum, that is the small brain. So here you're also going to, if you're trying to lift something, and so this is the also plane, this is not back and forth movement, but rather you're going up and down movement, which you're using also. You could also be using the movement, so you could also be using both the movement of the utricle and the saccule. Why? Because one, the head is moving downwards, not just going forward, but you're going forward and downwards, so the two follicles are also functioning, that is the utricle and the saccule. So here is an example of the movement. So you can see how you tilt your head, where you have the movement of the saccule, and then here you also have the movement of the crista ampullaris, which is the receptor cells found in the ampulla, and they are involved with the rotation of the head, or right tilting of the head, nodding of the head. All right, so here the information coming from the vestibular system, they leave the vestibule, and they come into they come into the pontomedullary junction, and so you can see some fibers as well crosses to enter the cerebellum, and then you have some fibers that ascend. So here you also have fibers that ascend, and then you also have fibers that go down to the body as well. So these are fibers that go down to the motor system to control your muscles, to keep put you at balance, and then you also have some fibers going upwards from the utricle, they go upwards. Okay, so this is also the vestibular nuclei in the medulla oblongata, and then you can see some of them going to the oculomotor nucleus. So the oculomotor is if you remember, is the third cranial nerve which controls the movement of the eye. This is very important because these also control the reflex pathway of the eye. This is called the vestibulo ocular pathway, because sometimes when somebody push it, immediately what happens, your eyes tends to be focusing on that area, and it helps you in your balance. So that is why you have the vestibulo-ocular pathway for the reflex, and reflex, assisting the reflex balance of your eye and your body. So here you can see the arterial supply of this is coming from so you have the anterior vestibular artery, which is a branch of the basilar artery. So the basilar artery is the union of the two vertebral arteries, and this is the basilar artery. So you can see several pontine arteries, but you also get to see a branch coming off from the basilar artery, and this is the anterior vestibular artery, which goes there to branch into two, and then you can get to see the common cochlear artery, and then you can also see the common vestibular artery going to innervates them. And also you have the venous drainages as well. The venous drainage is also draining into the venous sinuses. So it is via the inferior cochlear vein and the vein of the vestibular aqueduct. They drain into the sigmoid sinus. So the sigmoid sinus is at the junction, and this is because they are closer to the temporal lobe. So they drain into the sigmoid sinus. The sigmoid sinus is the curved one here, and then in this one here, as you have the sigmoid sinus will curved and continue down as the internal jugular vein. So a clinical application you have of the inner ear, you have the tinnitus. This is the constant ringing at the back of your ear. It feels like buzzing, ringing, sometimes clicking sounds. And there are several causes of this. This could be age related due to hearing loss, or in constant exposure to very loud noise, or earwax build up in the ear. Sometimes it could be infection and etc. So sometimes it could be medication, head injury, and sometimes it could be associated with other side or brain or neural injuries. And so treatment could be used. One could have a hearing aid, and you could also have the tinnitus retraining therapy, where you'll be trained to just focus on a particular sound to reduce unwanted sound on the background. And also go for sound therapy. You could do medication. You could change lifestyle to reduce the loudness of the voice. And these are more. And thank you very much. If you have any question, you can share it. I will send it to the WhatsApp group. All right, bye.